So this is video 2 of week 6. We have seen a few questions on the relationship between language and the mind, or eventually language and the brain itself. And now I want to just give you an idea of the kind of methodologies that we can use in the pursuing of answers to these questions. So the first one is going to be brain imaging. Brain imaging is literally looking at the brain and see how it functions. And if you want to know the relationship between language and the brain, so you look at the brain while the person is using language. Now, if you've never looked at a map of the brain, I'd really recommend it to you. And I have included here a couple of links that you have in the PowerPoint slides. Do click on those links so that you get familiarized on the different regions that are involved in the different biological functions and cognitive functions of the brain. Those are fun to look at. And while we don't have a complete map of the brain, or we don't quite understand exactly how the brain works, but we know enough to make it already a very interesting source of knowledge about the topic. Okay. So when it comes to multilingualism, one of the popular questions is to find out how the second language, meaning the language that you have not learned from birth, is differently represented in the brain from the first language, right? And how its development interacts with the cognitive landscape of the mind. For that, what you do is to identify the localization of the cortical areas used when solving tasks in each of the languages. So you have your subject, and then you make them solve something or even speak in the language that they learned when they were babies, so to speak. And then you look what areas of the brain are involved in that while the subject does that. And then you compare that to the same subject speaking or solving a task in a language that they have learned later on. That's what we call a second language. So then you compare to the areas of the brain that are involved in that second case. And then you see, by deduction, you see whether the first language is represented in a different place in the brain, or different structures of, well, properly speaking, different structures of the brain are involved in the functioning, in the enacting of the first language as opposed to the second language. The research motive many times is to determine qualitative changes that might ensue from the end of the periodic critical period. And that's why we looked at the critical period hypothesis, right? So a lot of these researchers, they want to know whether after a certain moment in time, the language that you acquire is treated differently by the brain. And that would be the critical period interacting with brain imaging. Okay, so that's what it's important. Now the methodologies this is a pretty recent, relatively speaking, pretty recent methodology, because technically we didn't have a way of looking at the brain before we invented the machines to look at the brain. So, but recently there has been a lot of excitement over the possibility that brains could be observed and observed in real time. And that cognition could be made visible by observing the brain, which is where cognition presumably happens, and that representations could be revealed. So that by observing where the action is happening, that you could actually deduce where cognition was embodied in the brain. Now, there are several methodologies. The first one is the ERP, the event-related potential, that measures electrical activity in the brain when the brain functions. And then there are two more methodologies that measure blood flow to the brain, because when the brain is activated, in a certain part of the brain is activated, the cells in that brain need a lot of energy because they're working. So they need, that energy comes from food that is transported to them by blood. So by looking at blood flows, in that particular area, we know that that particular area is operating. And we have positron emission tomographies and functional magnetic resonances that measure that blood flow. Okay, so we have you can measure either electrical activity of the brain or blood flow supply to certain area of the brain. Now, the first one, when you measure electrical activity, you have the advantage that you have an accurate measurement of the intensity and timing of the cortical activity. So electrical activity happens as soon as that area is working. The problem is that there's poor localization of the activity because once electricity is hard to contain. So once one area is activated, electricity has the tendency to spread to other areas that might look like they are also activated, but it might be just a side effect of the area, the area that is actually needed for that particular activity. So that is the ERP characterization. Now they're getting much better at controlling, at measuring what is true electrical activity related to a task that they are targeting from site electrical activity. But still, it is a little bit of a problem. Now, for blood flow, you have accurate localization because only the cells that need the extra energy because they have worked will be supplied with extra blood. But of course, there's poor timing information because the blood takes a little bit to reach the relevant cells. So it might be that by the time it goes there, it's also feeding on, taking advantage of the trip, so to speak, to feed other cells that were also in need of energy, but that were not related to the task that you are measuring. Okay, so one has accurate intensity and especially timing, but poor localization, and the other techniques have accurate localization, but poor timing information. So it's not perfect. Nevertheless, we have managed to identify certain areas of the brain that are involved in the different cognitive abilities or in different tasks. It's hard to make a diagnosis because this is what the images look like, which is, they're localized, but as you see, it's hard to pinpoint to what degree they are involved. Also, there was recently a little bit of a methodological problem with codifying such activity in colors because there was not agreement with the color scheme used and so forth. But that's pretty much where we are and what we work with. And neurolinguistics is advancing tremendously in today's world. So they are solving these problems towards a better understanding of how our brain works and how the different cognitive abilities are embodied in the brain. Now, most claims are based on the assumption that the first language is localized in the left hemisphere. This is one of the results. Although that's generally true, that language is in the left hemisphere, it is not absolutely or universally true. Lateralization is now considered to be a relative attribute rather than a categorical one, so that an individual is described on a dimension of lateralization. Okay, so it is true that roughly language is on the left hemisphere, but we see a lot of language in the right hemisphere, and we actually see that not only not completely constant across individuals, right? So there are people who kind of store their language more towards the left or more towards the right. And what's important to understand is that this lateralization is not something categorical, but that is a tendency. Okay, the same way that anatomically with everything else, there's also a tendency. I mean, we all have our heart located more towards the left part of our chest, but there's some people who have it actually very much in the middle, and even some people who have it more towards the right, curiously. So there is not, one thing we have understood is that even though you can have these maps of where language is stored, there is no universal claim to them, and individuals do vary. But what we do know is that irrespective of hemispheric lateralization, all these people learn and use language in ways that are functionally indistinguishable, suggesting that the precise cortical configuration may be less important than we might have imagined. Okay, so yeah, localizing language in the brain, in the brain map, and knowing where language is located has its interest, but it might be that it is not determinant of functionality. So it might be that there's more than one place in the brain that we can use to specialize on language, functionally speaking. And we know that also because people who are unfortunate enough to have had a stroke or an accident and they lose language, they might reuse other parts of the brain for that purpose, for developing language. We also know that people who are blind, for example, they might use centers that are in people who can see, that are used for seeing, and the blind people might reuse those parts of the brain to develop other abilities, even linguistic or a heightened sense of smell or even of hearing, right? And we also know that, for example, deaf people who are who cannot use who cannot hear, they use centers of the brain that people who hear use for hearing. They use it for spatial configurations or for motor and perceptual abilities, right? So the brain does have that neuroplasticity that we have learned about. So knowing where language is in the brain is very interesting, and but it might not tell us, it might not be the last word about the functionality of the localization of language. Okay. Now, more here about more links about language and the brain, and I invite you to click on them because they are excellent resources. Now, I want to finish with a note of caution, and is that many of the studies suffer from a lack of control over essential factors such as language proficiency and method of language learning, especially for a second language. So results can be also without consensus, so designs can scarcely be compared. Of course, we have people who specialize on neurology, but might be a little sloppy on these other in selecting their subjects. We know how complex the concept of being multilingual is, and if they just don't agree, one study doesn't agree, or one team of researchers that does not agree with the another team of researchers on who to count as multilingual, then how comparable their studies are is questionable. The same way as it's, people learn languages in a very different way, so experiences of language learning vary. And if one study does not have the same criteria in selecting their subjects as another, the problem that we have is that comparison between studies is made difficult, and when you cannot compare within the studies, then you cannot build up our knowledge about the topic. So many times sample sizes tend to be extremely small because it's very expensive to make brain scans of and to of any kind, electrical or blood flow scans, and then so you can do it only because you have the money that you have, and the people who allow you to look at the brain that you have. Many times you have sample sizes of 7-10 people, and then to generalize, given that people do vary so much on lateralization and on localization and so forth, it's a little questionable. On top of it, experimental tasks between studies are virtually never the same across those studies. So, of course, then if you have different experimental tasks, because each team of researchers comes up with a better test than the previous one or the next one, then it's difficult to put all those studies together and start learning something conclusive about neuroimaging and the representation of language in the brain, and especially of multilingualism, of the second language acquisition category. And then also the proficiency levels of the subjects are rarely controlled. So sometimes people who are bilingual from birth are in one study, and then another one from birth, I mean from babies, and then they are put to another study just takes heavily sequential bilinguals. Sometimes in that multilingualism continuum that we have seen, the studies do not control that all of the subjects either belong to the same, more or less same area of the continuum, or that actually they compare to other studies in the same continuum, and so on and so forth, right? So because there is not a unified criterion for all these variables that we have seen in the previous weeks, the results are all not that conclusive, right? I don't want to say with this picture that neuroscientists don't have any idea what they are doing. They do, but when it comes to looking at something as complex as language, and when it comes to looking at the further complication of multilingualism, they haven't been completely coherent and unified. So we know very little. So what do I say as a conclusion? I'm gonna take the same perspective as Bialystok took, which is, well, the question of okay, can images of the brain, even highly sophisticated ones, provide the necessary detail for determining how the brain is involved in something as multifaceted as language? Can we learn something? Can we learn anything at all from looking at the brain when we're looking at such a complex human ability? And she says, well, even if such information were available, which, as I've told you, so far it isn't much, she says, what would it tell us about how humans learn and use language by looking at the brain and having all these pictures of where the language is? And she said something that I think still holds. And she says, there is a dangerous reductionism at play here. The allure of observing the brain might lead one to conclude that one has also observed thought. Okay, so we must treat this new technology with both respect and caution, re-upping insights where we can, but remaining sanguine about the limits of its revelations. Okay, so yes, we see how the brain works. We see how the brain reacts or is working at the same time that thought and other mind abilities, mental abilities are operating. But to say that by having a map of the brain, we have a map of the mind, that is a jump that we have to be very cautious about making. So just up to here about this technique of brain imaging and how we can learn something about the relationship between language and mind by directly observing at the organ that we think is responsible for both.